Good morning everyone. So today let's discuss about how to solve the questions or problems in the Mendelian inheritance. Now, while solving the problems, actually what happens? We often find very much difficulties to how to find out the gametes, how to find out the offsprings, what will be their genotypes, phenotypes, isn't it? So let us study some basic formulas which will help you definitely to solve the problems. But beforehand, I would like to tell you that whenever you go through the question, you are studying the question, very properly read it, what is being asked there, okay? Then whatever is there, you just in the rough part just write it what things are being given there. If suppose the parent genotype is given, write that. If the parents phenotypes are given, so according to that what will be the genotype, so like that type of things, first of all you just frame it out in your copy, rough paper, then only you will be able to solve the problems, okay? Because the problems actually whenever it is given, the questions are very big but actually their answers are very small. So very patiently you have to read it first and then only you can solve it. So first of all, let's see the formula for the number of gametes. Now you see here, number of gametes, this is represented by 2 to the power n. Now n represents the number of heterozygotes, okay? So for example, you see here, in this is the genotype of a particular offspring, AA, not offspring, parent, sorry, okay? AA, B, capital B, small b, capital C, small c, capital D, capital D. Again this one, capital A, small a, capital B, small b, capital C, small c, capital D, small d. Now you see here, we are telling that for finding out the number of gametes, n is the number of heterozygotes, okay? So what happened? Here you see the number of heterozygotes in this one is only capital B, small b, isn't it? That's why n is equal to n small capital C, small c. That means 2 heterozygotes are there, so n equal to 2. So what will be the number of gametes? 2 to the power 2, that is 4. Again here you see all the 4 genes are in heterozygote condition. That means all are here, the number of n is equal to 4. That's why the gamete will be 2 to the power 4. Got it? Very simple. Then number of phenotypes is taken out by the same formula, 2 to the power n. Number of genotypes, this n will represent whether the cross is monohybrid, dihybrid or trihybrid. Genotype is taken out by 3 to the power n. Number of Punnett square also 4 to the power n. Now see, we are dividing here monohybrid, dihybrid and trihybrid cross. So accordingly what happened? In monohybrid cross, 2 to the power 1, gamete will be 2. Dihybrid, okay, trihybrid, then here the number of phenotypic ratio, similarly genotypic ratio and number of Punnett square, all these are mentioned here. Got it? Okay, now again what happened, we have studied about the bar bodies, isn't it? So bar body formula, to take out the bar body, the formula is that the total number of chromosome minus 1. Got it? Because we have already studied that number of bar body is less than the number of the X chromosomes, isn't it? So here what happened, from the total number of X chromosome we will minus 1. So suppose the question is given the number of bar bodies in XXXX female. Options are given 1, 2, 3, 4. So number of bar body will be what? Total number of chromosomes 4. Minus 1, that means 3. So answer will be 3. Okay, very simple these are. So you have to, again you see here, when a dihybrid yellow round pea plant is crossed with one of the parents, the progeny obtained is all yellow round only. What should be the genotype of the parent? Okay, so here beforehand very patiently study what is there, dihybrid yellow round. So that's why the phenotype will be, the genotype will be capital Y, capital Y, capital R, capital R. Got it? Then it is crossed with another parent. Now we don't know about the next parent, what is it, isn't it? So here again, but the again F1 hybrids, it is telling that the genotype is, or the phenotype is yellow round. That means capital Y, capital R will have to be present there. Now here 4 things are given, isn't it? Capital Y, capital R, small r, capital Y, capital Y, capital R, capital R, capital Y, capital Y, capital R, small r, and small y, small r, small r. Now this is a very simple dihybrid cross. So here the parent will be small y, small r, small r, because here these will show all. No, they will not show all yellow round parents offsprings. Okay, that means some different type phenotype will also occur. That's why here, for the appearance of yellow round in all organisms, the parent has to be small y, and small r, small r. Got it? Again, in the next question, you see a tall pea plant with round seeds has crossed with a dwarf pea plant having wrinkled seed. What can be the possible phenotypic ratio of the resulting progeny? Okay. Now here what is asked? A tall pea plant with round seeds. That means capital T, capital T, capital R, capital R. Crossed with a dwarf pea plant with wrinkled seed. Small t, small r, small r. Okay. Now, what can be the possible phenotype of the resulting progeny? Now, here you see, for the parents to be kept tall and round, two type of genotypes can be obtained. Either it may be capital T, capital R, or capital T, capital R, small r. That means in heterozygous condition also it may occur. So, if we cross it with the recessive parent, then what happened? All the offsprings will be if in this cross the offsprings will be capital T, capital R, small r. That means all tall round. Or if we do such a cross, that means this is a test cross, isn't it? So we will obtain a ratio 1:1:1:1. That means here both these two, that means all tall and round, and 1:1:1:1 ratio is possible. That's why the answer will be B or C. That's why I am repeatedly telling, whenever the question is asked, very patiently read the question. Okay, only then you can be able to solve it out. Again, the next question. When a dihybrid tall pea plant with round seed forms gamete, what will be the percentage of heterozygous gametes formed? Now here see, it is a very simple thing. Dihybrid tall pea plant with round seeds, that means capital T, capital R is given, and percentage of heterozygous gametes is given. Now here see, the other parent is not given, only one particular parent is given. So here there is no any question of formation of gametes, so the percentage will be 0. Got it? Then in the next question, in a Mendelian experiment, what genotypic ratio will be obtained in the progeny resulting from a cross? Now this is the cross. Okay, so let's first write the cross. Capital Y, capital R, small y, small r, small r. Now you know how to find out the gametes, isn't it? So sequentially you first of all find out the gamete. Okay, then after finding out the gamete, now you see here 2 capital Y is obtained, so we will write just 1 capital Y, capital R. Small y, small r, this one. And from this type of offspring, only small y and capital R gamete will be obtained. So we will prepare a Punnett square, we will write it. So what happened? We are getting capital Y, capital R, and small y, capital R in the ratio 1:1. Got it? That means we have simplified it very fast. So everything has to be clear. The presence of mind is needed. So what to do, how you have to first begin it. Okay? In the next question, it is asked that in one of the Mendel's crosses, the progeny obtained contained 4 phenotypes in the following numbers: yellow round 2450, yellow wrinkle 2550, green round 2550, green wrinkle 2450. Okay, now this indicates, now you see here, okay, how to solve it very simply. Mendel cross, got it? Then here 4 phenotypes are found. That means it is a dihybrid cross where 4 different phenotypes are found. When we will find this different type of phenotypes? When the cross is a dihybrid cross. Okay. So here the ratio is given, isn't it? Now you see this and this is same. These two are same, isn't it? That means all these indicate that it is a dihybrid test cross, because only in dihybrid test cross we obtain a 1:1:1:1 ratio in the offsprings. Got it? Okay. Now see here, a double heterozygous tall and yellow pea plant is selfed. In the progeny obtained, what will be the ratio of homozygous dwarf and green offsprings? Here First of all, the question is asked that double heterozygous tall and yellow pea plant. If it is double heterozygous, that means a dihybrid cross, capital T, small t, capital Y, small y. Okay, we have got the phenotype of the parent. It is selfed, that means it will be crossed with the same parent. Now, very fast we will take out the gametes. Now here we have find out the gametes. Now see, capital T, capital Y, small y. We have got capital T, capital Y, capital T, small y. Similarly, small t, capital Y, small t, small y. Okay. After finding out the gametes, we will put it in the checkerboard. Got it? Now see, here we have placed the gametes in the checkerboard. Just simple a Mendelian dihybrid checkerboard. Now here the question is asked, what will be the ratio of homozygous dwarf and green offsprings? Now see, how we will get here dwarf offsprings. First of all, for dwarf, small t has to be present. So let us see where we will get the small t. This is a small t, isn't it? Small t, so this will be small t, small t, this will be small t. Again this will be small t, this will be small t. Because if capital T is present, small t cannot express itself, isn't it? That is a recessive trait. We know that. So here this will be 4. Again for green to appear, see here, this is a green, this will be green, small y, this is green, this is green. That means 4 and 4. Got it? 4 number of green and 4 homozygous dwarf parents offsprings will be produced. Got it? So this way we have to find out the number of offsprings. Okay, again you see a cross is performed between pea plant as follows. So here the phenotype is genotype is given. Got it? How many different types of phenotypes will be produced in the progeny? Now you see here the cross is a trihybrid cross, that means n equal to 3. So formula for phenotype we have already studied, 2 to the power n, so 2 to the power 3e. Very simple. Okay. How many different types of gametes will be produced by a plant having a genotype? This one. So here n is equal to 1, only one heterozygote is present, isn't it? So 2 to the power n, 2. Got it? Okay. Now we have already studied about the pedigree analysis, isn't it? While we are studying the pedigree analysis, we obtain 4 different type of charts. That means it may be either dominant autosomal, dominant X-linked, recessive autosomal, recessive X-linked. So whenever a pedigree question is there, it will be definitely asking about what is the type of the what is the type actually, whether it is dominant or recessive, autosomal or X or sex linked. Okay, so there what happened, there are some important points to how we can find out this pedigree chart. Now see, for being an autosomal dominant, they do not skip generation, very first thing. Okay, and here both the parents are affected but children remain unaffected. But always remember, sometimes what happen, 1-2 points may be here and there. That means sometimes they may not fulfill all the requirement, but still. Now see here, don't skip generation. This is the first generation where we are seeing the occurrence of disease. Second generation, third generation. That means all the generations are showing the disease. So this is an autosomal dominant one. Okay. Now autosomal recessive, they skip generation. Unaffected parents have affected children. Now see here, the parents are unaffected, the children are affected, and this generation is being skipped. So this is the autosomal recessive one. Got it? Then X-linked dominant, here the disease never transfer from father to son. Now see here, the father is affected but it is not transmitted to the son, it is transmitted to the daughter, and all daughters of an affected father will be affected. See here, from this parent 2 daughters are formed, both are affected. Got it? So this is the X-linked dominant. So main thing you have to remember these points. Okay, then only you can be easily able to solve it. Then X-linked recessive, here the male are more affected. See, maximum males are affected. The disease transfer from mother to son and father to daughter. Never it transfers from the father to son. Now here you see, if the disease is occurring in the son, that means the mother has to be a carrier. Okay, the disease is not expressed but she is a carrier. So here it is transferred from the mother to the son, from the son to the daughter. Okay, that means a crisscross pattern is observed here. So these are the main points for determining a pedigree chart, whether it is a dominant or recessive, autosomal or sex linked. Got it? So remember these points and accordingly try to solve the charts. Okay, and the most important thing, repeatedly I will tell you, very patiently read the question what is asked. Okay, and simultaneously just draw it or write it in the rough paper and very simply solve it, and definitely you will then able to find out the correct answer. Hope this video will be helpful to you for solving out the different genetics problems, and still if you find any difficulty then please ask me.